Okay, so the last little bit of technology that I want to talk about is so-called short-read sequencing. This is really what's driving a lot of genomics and bioinformatics right now because it has sort of supercharged the ability to read out the secrets, blueprint of life in the form of the complete genome sequence for any organism that you're interested in. I mean, you can sequence a human genome basically in a day, and for relatively little money as well. So what this really does, to summarize it very, very briefly, is that you can only read short little pieces at a time. So what you get is an enormously large number of these so-called short reads. So it's sort of like taking, the genome sequence, shooting it with a shotgun that smashes it into tiny little fragments that are on the order of, let's say, 100 to 150 letters in length. You start out from maybe a chromosome that might be 200 million letters in length, and you're dropping it down to lots and lots of tiny little pieces that you have to put back together again. So I think you can see right away there's computational challenges involved in inferring the complete genome sequence from all these short reads. We'll talk a little bit about that later in the course. But right now, let's just say that we're generating huge numbers of these, and each individual read is short. So typically, when we're trying to put Humpty Dumpty back together again, we are, in biologist terms, aligning many individual short reads so that basically the overlapping short reads are lined up to each other such that they're reading the same piece of the sequence. So traditionally, people would show kind of a horizontal axis that represents the coordinate in the genome sequence from beginning to end, say, of a chromosome. And then underneath that, showing individual short reads that align, in other words, read or cover that particular portion of the genome sequence. So here are some example views from what that looks like in a so-called genome browser viewer. As I said, the horizontal axis here represents position in a piece of genomic sequence. This is zoomed in very small, so actually that length here, let's say that's 100 nucleotides in length. So each one of these little lines is a single short read, okay? And so what this display is doing is it's simply stacking up all the individual short reads that have been aligned to the so-called reference genome sequence. So the horizontal axis here is the position in a inferred or known genomic sequence, and then underneath it are the actual experimental short reads that we're getting from one of these sequencing machines from one specific sample that's been sequenced, okay? And so you can see that at some positions, maybe we have more reads stacking up. Maybe in other places we don't have as many reads stacking up. That might just be sampling error, just due to the fact that this is sort of a random shotgun sampling process. Or it might reflect some real differences in the sample. For example, if these short reads were being generated from RNA that represents so-called transcribed genes, what's that going to do? It's basically going to pick out just the regions of the genome that are actually transcribed, in other words, that are genes. Because as you may have inferred from the numbers that I told you last time, the human genome is a very big place, but actually, relatively speaking, there aren't that many genes, and they're fairly short compared with the size of the human genome. So actually, huge portions of the human genome are not, quote-unquote, protein-coding genes, and are not expressed at a high level. So this vertical axis represents a number that biologists refer to as coverage. the simple definition of coverage is in a given experiment, it is the actual number of observed reads that cover or align to a particular letter position in the genomic coordinate system. Okay? So here's a region that appears based upon the short reads generated from RNA to have a high level of quote-unquote expression, in other words, transcription of this region into RNA. Whereas here is a region that doesn't appear to be transcribed into RNA at all, because we're just not seeing any experimental reads from that. Questions about the terminology? Sorry, I didn't quite hear that. Could you speak up a little bit louder? Right, right. So literally, you could think of that as just being an integer that starts at 1 and goes to 200 million if your chromosome is 200 million letters long. The short reads, yeah? Yes, exactly. So the coverage at any given position is defined as the number of reads that are aligned to that specific position in the genomic coordinates. It's just an integer. Question?
As you go lower down on the y-axis, does that mean that those letters, those strings are later on in the location necessarily or not?
So the layout of these short read visualization, you know, there's algorithms that just try to pack it in as kind of efficiently as possible. I wouldn't attribute any particular meaning to the fact that this guy's in the bottom and this guy's at the top. What matters here is the horizontal coordinate gives you basically a column that represents a single nucleotide letter in the genomic sequence, and all of the individual reads that read that particular letter from the source material that was put into the sequencing machine.
So what does enrichment really mean in this context? I saw that a lot in a lot of papers, but I don't really know what it is.
Well, basically in this view, you should think of the vertical axis as just being the coverage value, and the horizontal is the same coordinate system for the genomic coordinate, but actually zoomed out to a larger scale. So, you know, that might be, let's say, now 1,000 or some larger number of nucleotides representing kind of a gene region, say. Okay? Did I answer the question? I'm not sure if that was what you were asking.
So the enrichment actually means you are zooming into only a particular...
This is coverage. This vertical axis is coverage. So maybe something that I'm failing to say is that the source material, you could think of it as just being a pool of randomly fragmented DNA, okay? And you can imagine we're just like, you know, a classic statistics problem where you reach into an urn and you take out a ball, or a bag, you take out some random object. We have a sampling process here that's randomly sampling those short fragments out of this big mixture, okay? So in that kind of a random sampling process, which we're going to talk about explicitly as we go into the probabilistic models, basically the mean coverage that we would expect will be proportional to how much of that particular sequence, piece of DNA, was present in our original sample. So it's basically linearly proportional to the relative amounts of DNA in the sample for different regions on this coordinate system. And to make a long story short, if you're studying, for example, gene expression, which is just means level of transcription, some genes produce a lot of RNA, other genes produce no RNA at all because they're turned off. Okay? So what's being measured here in this so-called enrichment is basically the level of transcription of a particular gene compared with neighboring regions that are probably not encoding a gene at all. This is probably just the level of random noise in this analysis. Another question.
Is the input to this process RNA?
So let's say, for the purpose of argument, since I've been using RNA as the example all throughout here, that yes, the input to this was an RNA sample, so-called messenger RNA. There's actually different forms of RNA. But messenger RNA is basically the form that is produced to make proteins. Okay. So in a typical kind of short read data set, people would probably be aiming for a coverage of around 70x. So 70-fold coverage. It basically means the number of reads stacking up at any given position along the genome would be around 70. Okay? So you're reading the same letter in the genome around 70 times from your sample.
This is for DNA sequencing or RNA.
Well, it really depends upon the kind of experiment you're doing. I mean, I've had some where we shot for substantially lower. It depends on what you're trying to get out. So, but this is probably pretty typical for both transcriptomic RNA and genomic DNA experiments.
If these were made from RNA, why would the non-genes have any fragments?
So this is actually something there's a lot of argument about. Some people basically insist that because you can detect some signal sort of everywhere, that something like 90% of the human genome is transcribed. But this is a complex experimental process. There's lots of sources of error that go into this, including the biology itself. I mean, there's no law that says that biology has to be perfect. I mean, are any of us perfect? We do things perfectly. We're talking about very complicated molecular machines, and if they, sometimes land on a piece of DNA that doesn't do anything in a traditional protein coding gene sense, and they make one RNA out of billions from that particular position, does that kill you? Not necessarily. So evolution doesn't care if that small amount of leakage happens. So the bottom line is there is noise from many, many, many sources in this kind of an analysis. The interpretation of that noise is sometimes a challenging and controversial scientific question, but you can see the kinds of numbers that we're getting. You can see there's pretty clear signal there. I mean, you can see there's background noise, and we're clearly way, way, way above the background noise when we hit that gene region. Question?
So does that mean some genes could convert to RNA, but finally not end up being a protein?
So there's all sorts of things happening in biology, and the bottom line is, almost anything that you can ask, yes, biology will have, some special case where it will do that kind of funny thing. So yes, there are regions of the genome that produce RNAs that do not make proteins, yet which can be shown to do interesting and useful things through the RNA molecule itself. RNA has interesting properties. Oh. How do you estimate the coverage? Well, let's start with the completely vanilla average coverage value. Let's say that I have, I have 10 million reads, okay? And I have a average read length of 100 nucleotides. So 10 million times 100, I'm now at a gigabase of read nucleotides, right? Total. If I'm sequencing a genome that, let's say, was only keep it simple, let's say it was 5 million nucleotides in length, then my average coverage is simply my total nucleotides, which we said was a gigabase, divided by my genome coordinate system length. It was basically going to tell me what's the average number of reads hitting any given position. So in that case, I'd have an average coverage of 20, right? So that's the basic idea. It's simple stuff. Okay. Thank you for asking all these questions. This is really great.